everybody. This is Matthew, Vrushali, and Michael here to talk about our microfluidics project. The goals of our project were threefold. They were to create a fluid-functional microfluidic device using the tools available from CIDR. Also, to develop a program which can parse a user-entered script and control a biological experiment on a microfluidic device, as per the script's description. This would allow automation of the experiment and provide consistent test-retest reliability. Finally, the third goal was to come up with additional primitives that the lab can make use of as building blocks for more microfluidic experiments.
For the primary microfluidic device, we use the 3DUF tool to generate our fluid functional device. Seen here is the selection and parameter setting used to build the device in both the flow and control layer using components such as channels and ports. The parameters are set such that it will work with the CNC mill that we are currently using. At the end, the data is exported to OtherPlan for both tool planning and controlling the desktop CNC mill. The primary file type we are using is SVG. Here is a similar device as that one designed in 3DUF using the Mint description, then an automated place and route completed by Fluigi. Here is the final output from the Mint description. You'll note that some of the ports are rather close and may not work with our CNC mill. Therefore, the final design that we went with was the original one seen in 3DUF. Here's the flow and then the control layer. What you see here is the first step in fabricating our microfluidic device on the other mill CNC in the CIDR lab. At this point in the fabrication, the microfluidic channels are being milled. You'll see here that there's an accumulation of plastic indicating that the drill bit has become dull. This was a common challenge that we ran into while manufacturing the devices, given that they have a lot of complex parts. With the flow layer complete, we now move on to the control layer. Here we see the ports being drilled out. And we can also see on the computer how far the depth is completed in the milling process. At the completion of both the flow and currently the control layer, the device is cut out so that it matches properly. With the device completely milled, the ports are tapped so the fluid lines and syringes can be attached to both layers. Both devices are cleaned and matched up to make sure that everything was done properly. Looking at the devices under a microscope gives us an idea if the milling was done as expected. Looking closely, we see a small flaw in the milling here. This was especially evident when we looked under the microscope, showing that the tool did not go completely through this channel. It was more apparent when we looked back at the milling and actually noted that with this particular plan it did not go as intended. Adjustments to the mixer, such as in some cases assembling directly using the channel primitive, resulted in better tool planning. There were additional challenges in milling as seen here by the various parts that had to be replaced during our iterations, namely several drill bits and a pulley on the CNC mill. With the final device ready, the connections and seals are attached so the tubing and syringes can be connected. The layer between the control and flow layer, etched in polycarbonate, is a thinner layer of PDMS. With the layers aligned and the PDMS between, binder clips are used to hold everything in place. The entire device is then placed in a vacuum chamber to remove any bubbles in the layers. The 5 syringes seen here control whether a valve is open or not. The syringes pulled back pull the PDMS layer away from the flow layer and allow fluid to flow freely. This is followed by the 3 inputs seen here, using basic dies to test fluid flow, control, and mixing. The overall flow rate is set by this dispenser, both set to a standard rate around 1.5 milliliters per hour for this case. The 2 outputs for this device on the left are open to dispense into a waste container directly. And now it is time to finally check that everything is fluid functional. The green die is started first to see how it flows through our device. It is also checked that it is entering the chambers designed for observation correctly. This is then coupled again with a closer view under the microscope, as seen here. Adding in the last 2 inputs allows us to check what happens with pressure changes in the valves, how well the mixers work, and finally what happens over a series of time if we remove the green input to see how well the mixtures change over time. With a microfluidic device now milled and is successfully tested, or at least fluid functional with components derived for cell observation that could be further refined later, we now move on to the next portion of our project. Our design was intended to be somewhat flexible in that it could be used with different protocols, such as ones that involve different mixture but still had a need for observation. Traditionally, microfluidic devices are manually controlled, but what if there was a way to automate them for ease of use and reproducibility? Let me now introduce our tool, an extension of Neptune, called Percy.
Here are some snippets of our code. This is our main script which parses the control commands typed in by the user. We are parsing the script using regular expressions in JS. There are lots of useful methods that can be used to detect keywords like open, wall, push, pull, etc. in the script. For example, the test method returns a true value if the keyword exists. Another example would be the search method, which returns the starting index of the keyword, which is a very vital part of the script, as the keyword's search index uses a spatial logic to detect the words. The script also includes a syntax check logic, which is limited to checking whether the user has typed in the correct words or not. The script stores the JS objects in an array, which enables the reusability of the object data. In order to keep track of the volume initially pulled by a dispenser and the volume remaining in the dispenser after a certain push operation, we have implemented a search algorithm in which if a previous dispenser object has the same port as the current dispenser object, then it calculates the current state of the object by taking the remaining volume into consideration. Each control command is then executed asynchronously using the set timeout method, which makes it easy to schedule and run multiple commands at the same time. The method also allows the script to introduce a user-defined delay in running the control command, thereby helping in the scheduling process. The data object is then sent to another script which converts the control commands into actual hardware computations. The script calculates the PWM values for the servo motors and the relative displacements of the dispenser pumps. As you can see, there is another asynchronous command embedded in here. This basically divides the execution time into millisecond frames. Each frame has an equivalent PWM value. The set timeout method here mainly schedules the time until which a control command should be executed by sending it to the Arduino board. The code also detects the invalid volume dispensing conditions, and an error message is sent out to the user in such a case.
I'm going to give a quick example of how to use Percy to control a microfluidic device experiment. We've opened the web page and we're going to click the try button here, which opens up the automated scheduler. So here it says to enter your control script and then sit back and relax while Percy automates your experiment. And the first example I'm going to show you is what happens when an incorrect syntax is used. So here we're going to type in @0/3. Instead of writing pull or push here, we're going to write dispense. And we're going to say 40, semicolon, run. So the page says check syntax, which means your syntax was incorrect. What we're going to show you here is a correctly working one with an initial time of 0. So we're going to say @0/2, pull in 1, 40 microliters. So we're going to click run here, and at time 0, over 2 seconds, we're going to send a command to input 1 to pull 40 microliters of fluid. Finally, we are going to run a microfluidic experiment using Percy with the correct syntax, integrating asynchronous timing. So I'm going to paste some code in here. We're going to say @0/10, open valve 1, @2/1, pull into 40 microliters, @4/10, open valve 3, and @6/2, pull in for 30 microliters. Remove a space there. And we're going to click run, and at 0 it'll open valve 1, at 2 it'll pull in 1 for 1 second, at 4 it opens valve 3, and then at 6 it pulls in for 30 microliters.
Wrapping up, here is an example of the device that would be interfaced on a local level through socket.io using Percy. For future work, Percy can be expanded with integration into Neptune. Going briefly back to our microfluidic device, at the conclusion of our project we identified a few points for further development. First, we saw potential in adding multi-level primitives that could assist with air bubble capture on initialization, and finally improvements to cell visualization by components with areas less likely to capture cells.